chromatic responses to V4 but not V3A map onto human behaviour. So, as I said in the title, I'm going to look at V4 and V3A using fMRI techniques and their response to chromatic and achromatic stimuli. So firstly, what are these areas? We have here a picture of the brain, looking at it from side on. From the very back here of the brain, we have what we call primary visual cortex, or V1, and there are two visual streams coming off it. The dorsal stream going along the back towards the top of your brain, and the ventral stream going here towards the temporal cortex. The dorsal stream is often called the where pathway. It's implicated in vision for motion, like grasping and other quick visual judgments. The ventral stream, however, is often referred to as the what pathway, and it's implicated in recognition and fine discrimination. The aim of both the experiments I want to talk to you about today was to examine the colour-specific mechanisms in visual cortex. So we chose to look at V1 alongside one area from each visual stream. So this is the ventral stream, and we've twisted the brain round here, so now you're looking at the bottom, and it goes from V1 all the way to V4 here. This is the dorsal stream, and so we've twisted the brain back round again so that you're looking at the back, and this goes again from V1 to V3A here. These are both good areas to look at because they share the same topography and hierarchical position, both being located next to V3 here. You can see V1 here is at the start of both of these streams. So consistent with the what and where pathway idea, V4 has for a long time been considered specialised for colour, and V3A is most commonly associated with motion selectivity. So firstly, why is it difficult to look at colour-specific mechanisms? The spectral properties of stimuli are changed within the eye as a result of spectral absorption in both the lens and macular pigments. For the lens, the spectral absorption varies as a function of age. This graph shows the changing spectral absorption properties over the lifetime. On the y-axis, we have optical density, which is just the log of absorption. On the x-axis, we have the visible spectrum from 400 to 700 nanometers. Each line is a person of a different age, with the arrow pointing up as participants get older. As we can see, the spectral absorption properties of the lenses in the eye vary systematically with age. This particular issue can be overcome by testing young age-matched participants. The picture on the right shows the macular pigment density across the macula on the retina. As you can see, absorption varies over this area, and this also varies across individual participants. So within individual participants, the spectral absorption varies in the retina depending on the morphology of different cell types shown here. For example, in the fovea, cones are longer and thinner, where in the near periphery, they're shorter and fatter. Taken together, all these variations mean that specifying visual stimuli to stimulate colour-specific mechanisms is a challenge because the spectra absorbed within the eye varies so greatly, both within the individual eye and across different eyes. This issue is particularly salient for stimuli that cover a large amount of visual field. So, why is it particularly hard to study colour-specific responses in fMRI? In fMRI, we routinely present suprathreshold stimuli over large areas of the visual field. This is so we can increase our signal-to-noise ratio and find a bold response. In this study, we wish to probe the characteristics of colour-specific mechanisms. However, when we increase the visibility of our stimuli well above threshold, we are more likely to encounter leakage into another domain. For example, if we increase input into the L-M pathway, we may be inadvertently adding luminance. This will be a big problem if mechanisms in similar areas to the ones we are looking at are sensitive to this leaked signal. Sorry, that's me showing the leakage that could go into the luminance domain. In order to solve this problem, Barber attempted to saturate the luminance-sensitive mechanisms using a flickering luminance background, which looks similar to this. The theory behind this is that the luminance system will be responding so strongly to the flickering changes in luminance that when we add colour, it won't be able to see any additional leakage into the luminance domain. In their study, they concluded that this random luminance modulation method can allow extraction of chromatic signals independently of the confounding effects of luminance. The current study will examine whether this method can be effective in fMRI as well. So the first question we want to ask is, does the random luminance modulation method work for fMRI research? That is, can we use it to elicit colour-specific responses in visual cortex? One way to check this is to check whether the responses in the brain to the two components of the stimuli, that is, our randomly flickering background and the target colour we're going to overlay on top, are linearly additive. We will test this by having various levels of contrast of both the background and the overlaid colour, which I will show you in a moment. So our random luminance modulation background is at 5 cycles per degree and is flickering at 20 Hz, and the chromatic probe rating is 1.25 cycles per degree and it's changing phase at 1 Hz. The whole thing subtends 20 degrees of visual angle. So our question for our first experiment is, are we able to demonstrate linear additivity of the responses to the two components of the stimuli? This will do two things. Demonstrating linear additivity gives us confidence in the method, but also further examining this additivity, for example, in different color directions, we can look at the independence and interdependence of neuronal populations that encode for color. So this is the model we're going to use to decide whether our responses are linearly additive. I don't want you to worry too much about this equation, other than to say it's a modified version of the Necker-Rushton equation, which we typically use to model gain control. This first part of the equation is our model of the response to the achromatic luminance background. And the second part is a model of a response to chromatic target stimuli. What really matters here is the addition in the center. Irrespective of our response to the different components of the stimuli, if they are linearly additive, the two responses should add together. So here are our stimuli. We're going from 3% achromatic contrast up to about 50. And we start this first row with no color added, and then we go up to around 3% contrast. And this is in the L-m dimension. We displayed these stimuli in an event-related design, a total of 20 times each, to our participants in the MRI scanner on 2 separate sessions. So if I twist this around like this, this is how my first graph will be shown, just to orient you. So achromatic contrast going up in this direction, chromatic contrast going up in this direction. So here are our results. We have our 3 areas, V1, V3A, and V4. So if I just bring your attention to the V1 graph here so I can explain what these graphs represent. So as I said, we have achromatic contrast going up in this direction and chromatic contrast going up along this axis. Our percent signal change value for that area for that condition goes up along the z-axis. Black dots are measured percent signal change values, and the coloured surface you see is what the model I showed you before predicts. So we can see from these graphs, the model seems to be predicting our data quite well, particularly in V4 and V1. V3A, however, appears to have no or very little change in response to the increase in our achromatic background. Well, this looks very nice, but it's quite hard to interpret on these. So if we have a look at the graphs from a side view, so we're going to be looking both from the x-axis like this and from the y-axis like this. So these plots are the exact same data, but now in 2 dimensions. The top 3 graphs have achromatic contrast going along the bottom, and chromatic contrast goes up from green to red. The bottom 3 graphs have chromatic contrast going along the bottom and achromatic contrast going up from black to grey. Adjusted R-squared values are also shown in our top graphs. Again, the dots represent the measured value and the lines are our model fit. In V1 and V4, we can see our model has accounted for most of the data really well, particularly in V1. Notice the axis here has changed, so our values are higher in V1. In V3A, we see a highly compressive response to achromatic contrast to the point it looks completely flat on this graph. We suggest this may be because V3A is motion area, and even when we have this low 3% achromatic contrast, the random luminance modulation background is still a highly visible stimulus, so perhaps we haven't reduced the achromatic contrast low enough to see a lower response to the achromatic contrast. I also want to draw your attention to these points here. This is where our random luminance modulation background is the lowest and our chromatic target contrast is the highest. So these points elicit stronger responses from the brain in all areas than our model would predict. We suggest this is because at such low achromatic contrast, our random luminance modulation background is no longer working. That is, the achromatic contrast is not high enough to cancel out the luminance effects from the chromatic target, which is what we're using this to avoid. So future work should use stronger achromatic contrast than this. So we believe we've demonstrated independence of the random luminance modulation background and the chromatic target. This isn't too surprising as the temporal and spatial information in these stimuli are different, but we still feel this shows the robustness of the method. So we can now dig deeper into these areas and see where in the brain responses to colour align with behaviour. In terms of behaviour, the thresholds for being able to see the different colours are differently affected by spatial frequency. On these graphs, the y-axis is reducing contrast and our x-axis is increasing spatial frequency. The black line here is supposed to be the threshold for seeing your stimuli, but obviously they'll be different here depending on your screen. In the L-m dimension, we can see this increase in contrast which we require to see the stimulus when we get to high spatial frequencies. But in the S-cone condition, this effect is much stronger. So that means it's much more difficult to see high spatial frequency S-cone isolating stimuli. So for this experiment, we had 2 conditions, L-m and S-cone isolating, 2 color conditions, I should say. 3% contrast was added in the L-M dimension and 10.5% added in the S-cone dimension. Each was tested at 2 spatial frequencies, 1.25 cycles per degree and 2.5 cycles per degree, to probe this threshold difference and see if it holds in the brain response. Also, to ensure our random luminance modulation succeeded in saturating luminance-sensitive mechanisms, we also tested an achromatic condition, adding 15% contrast. Each stimulus was presented separately in a block design 3 times over 3 separate sessions. So we presented the stimuli like this: 15 seconds of just the background, 15 seconds with the colour added, times this by 8 presentations per run, with 3 runs per condition for 6 conditions and with 6 participants. So for some specific hypotheses, V4 has been consistently associated with colour vision in previous work, so we would therefore expect greater response to colour from V4 than V3A. With regard to spatial frequency effects, we may also expect V4 responses to fall more in line with behaviour, so that would suggest a smaller response to S-cone stimuli at the higher spatial frequencies in V4. So we found the average % signal change in each area in response to our stimuli. We then averaged across the 3 runs of the same stimuli for each participant individually. So this is how I'll present my data, so I'll take you through it bit by bit. So along this axis, we have the response to the lower spatial frequency stimuli. Along this axis, we have the response to the higher spatial frequency stimuli. So each dot is an individual person's score. And this bar graph here is just the mean for all the participants. This black line is a line of best fit. So a strong correlation on this graph means the participants' bold response to high spatial frequency stimuli was strongly related to their response to low spatial frequency stimuli. So for V4, we can see strong correlations between conditions for each participant and a robust response here to both colour conditions. In the L-M condition, we can see from the slope in the graph, there is very little difference in response when we double the spatial frequency. However, when we look at the S-cone condition, doubling the spatial frequency almost halves the response we detect from V4. In V3A, our correlations are not as strong and the responses to colour are generally lower. There is also a much lower effect of changing the spatial frequency in this S-cone condition. So responses are less aligned with behaviour in this area. However, we are still finding significant effects of colour in V3A. But did our random luminance modulation work again? That is, did we isolate chromatic signals? In the achromatic conditions, we didn't see very much at all. Here we can see the average % signal change for V4 and V3A plotted in exactly the same way as before. As we can see, there's not much going on here. Participant scores across spatial frequency conditions aren't correlated. We're working with very low and sometimes even negative signal change values. So essentially, yes, the random luminance modulation appears to have saturated any response to changes in luminance contrast. So from this, our random luminance modulation technique appears to have worked in both experiments, just in saturating any response to luminance. V4 does respond to colour more than V3A, as in previous work, and is therefore more likely to underpin our sensitivity to colour contrast. V4 also shows a strong interaction between spatial frequency and colour, while V3A does not. This suggests that V4 is more aligned with behaviour than V3A. However, V3A does show a strong colour response still. Takimura and their colleagues found that there are strong white matter connections between V3A and V4, and that their responses to stimuli are linked. Thus, the colour response in V3A could be partly due to this connection with V4. I'd like to thank you for listening, and thank you to my lab for all their help.
Great, thank you so much. That was, I'm just going to turn my timer off before it beeps rudely. That was perfect and really interesting. So I have a personal question while we wait for questions from the audience, while people think about, I can see some coming in now. Basically, it's about the visibility of your stimulus for the second experiment. I noticed you said it quite low in terms of the % contrast, I think.
Yes, so these images aren't accurate. I should have said that during the talk. They're not accurate to what they actually look like.
Oh yeah, that's fine. Sorry, I always do that myself as well. But I was wondering about whether it was set. I was assuming everyone saw the same stimulus, but did people? Yeah. It was super threshold then.
Yes. So this was it's surprising, but it was quite a lot above threshold. So the idea with the different percentages was that they were supposed to be equally visible. At least they were to me when I was a participant. So because you've got the flickering background on all the time, you completely adapt to it. So any change you will see, but you could see all of these stimuli quite clearly. In fact, the most difficult one was the achromatic one. It doesn't look like it on this, but it was.
Yeah, no, I can imagine. I think like, because I'm a psychophysicist at heart, I'm obsessed with visibility of stimuli and things. So, but that's great. Thank you. So we've had 2 questions so far. One of them was, what was the rationale for using slightly different stimuli across experiments? I don't know if you've partly answered that anyway, but.
Yes. So I don't know. I'm presuming that they mean because this here is A, square, and B, we have only used the central portion here of the square, whereas in the other within the other experiment we're using the entire circle has got the color on it. So basically this second experiment here was actually done first, so the stimuli more closely resemble what Barber did, because I wanted to base it off what they did. So yeah, it's basically just because we did this one first, and then we realized when we were doing the first experiment, which was the second experiment, that we would be better off knowing exactly our degrees of visual angle, which you can't do with a square, and it would be more retintopically accurate to do it that way. So it was just a practical thing.
Great, that's really nice. So we're almost out of time, but I do have one more question for you just before we let you go. Basically, you've made this sound like this was a check that this method would work for brain scanning and it's appropriate for differentiating between these signals. So that makes me think you've got plans for using it for something.
Yeah, so I probably should have got a picture of this, but the first experiment where I'm looking at linearity could be really interesting. I was pre-COVID looking at doing the same experiment in the S-cone dimension, and then the plan was, and still is, to have the achromatic background on all the time like it was in this experiment, and have the linearity that we're testing be between L-M stimuli and S-cone stimuli, so add those on top of each other in different levels. That would be interesting because we could say whether there were independent neural populations for each individual colour. So that would be really exciting, something we'd really like to look at.
Great, well, I hope post-COVID we can get some data from you to look at that. That sounds really good. But in the meantime, we do have some more questions, but I'll send them to you afterwards if that's okay. Thanks again for a great talk. That was really interesting. I'm really looking forward to seeing what else you produce.
Thank you.